We have our next speaker coming up to the stage, and I'm excited to introduce Dr. Greg Grover. Greg, thank you for being here tonight. He is the co-founder and CEO of Karios Technologies.
All right, let's get this fired up. Hi, everybody. My name is Dr. Greg Grover. I'm the CEO, co-inventor, and co-founder of Karios Technologies. The core of Karios' technology platform was some cross-linking chemistry I invented as a PhD student in organic chemistry at UCLA. I then took this chemistry down to UCSD as the American Heart Association postdoctoral fellow, and there I worked with bioengineers and cardiothoracic surgeons to understand problems more deeply, how we could use this platform technology to address unmet needs in surgery. And the first focus that we wanted to address with our technology were post-surgical adhesions. We view these as the holy grail of post-surgical complications because this is what surgeons have told us. 5 out of the 10 most common surgical procedures are significantly impacted by these post-surgical procedures or post-surgical adhesions, as shown on this slide. Now, there are 2 approaches to deal with these adhesions. The first is a solid film or a solid membrane, like Ceprafilm, for example. You can think of this like saran wrap. These films suffers from issue of dislodgement, rapid degradation, and they're also difficult to prepare and use. Imagine saran wrap covered in water while wearing gloves trying to wrap it around an organ. The second approach is to use a viscous hydrogel solution. So the concept here is you separate these 2 tissues so that they can't interact with each other. These gels suffer from high swelling, rapid degradation, and there's also issues with preparation at the surgery site and their use. And so the key here is that none of these approaches have been effective. And so reoperation is required to remove these adhesions. And so we first chose to focus on the heart. And here we invented Tissue Shield, a product that's designed to protect the organs and tissues. And our goal here is to protect these patients and save hospitals money. The illustration on the left shows our figure being used on a heart. And here we're going to show our prototype being used in a surgical setting. This is a partial sternotomy in our pig study. You can see that the prototype easily fits in the surgeon's hand, and it's easily sprayed over the surface of the heart, rapidly gelling and creating this protective coating. It's important to note that cardiac adhesion removal triples the rate of injury and death on reoperation for these cardiac patients. So we really wanted to focus on this area because there are not many solutions to address this need. The core of our technology is this novel 3-component hydrogel platform. The first component is this resin here shown in pink, this activated aldehyde that interacts with our hardener shown in red. This is what cross-links and forms the hydrogel. But what's unique to our technology is we have an added tissue binder derived from the muscle binding protein domains, DOPA. And so this reacts with tissue surfaces in the liquid state, forming a protective coating that when cures, prevents cell and proteins from binding. We've developed this dual chamber, dual syringe, ready to use solution as shown here at the bottom. When we were doing feedback analysis from surgeons, they said they wanted something easy to use that would require no assembly or preparation. We have great preclinical and safety and efficacy data. Here I'm showing figures from our Nature Communications paper from last year. The graph on the top left shows how we prevent cellular attachment to the formed gel. The graph on the top right shows how we're cytocompatible and not toxic to cells. The graph on the bottom left is from our rat study, where we show that we prevent adhesions in our cardiac rat model. And then the figure on the right is a histology slide where you can see that the material after H&E staining is still bound to the surface of the heart. Our technology is supported by 3 patents with exclusive commercial licenses from UCLA and UCSD and 4 peer-reviewed publications. We're also currently developing our own IP and growing our portfolio as well. So we have a proven prototype and a clear timeline. We were founded in mid-2020 based on the results of these rat and pig studies that we published with our prototype. And I mentioned 3 patents issued. We're raising a seed round right now of $2 million, where the key takeaways are to scale up our manufacturing for this prototype, this dual chamber prototype here, as well as performing a piglet study, which we just received a score of 25 on our NIH SBIR phase 1 grant. We'll also be doing FDA submissions. We have fantastic collaborators and partners, including leaders in this space, including Credence Med Systems, Lyophilization Technologies, and West Pharmaceuticals. Our goal is to get into clinical trials as quickly as possible, with the first focus is a first-in-human study of 30 patients. Our initial area that we're focusing on are children with congenital heart defects. This is because this type of patient has a staged operation where they require that second operation anyway in 3 to 4 months. Therefore, we can do a 1-to-1 study there. We also are looking at moving on to a pivotal study, but our goal is an acquisition by a strategic partner such as those shown here, J&J, Baxter, and Ethicon, for example. There are potential exits at each stage here after our clinical studies. We have an experienced and successful team. I personally have developed other hydrogel products, most recently as CTO at Contraline, where I developed another injectable hydrogel system to occlude the vas deferens as a male contraceptive device. We have surgeons, former entrepreneurs, as well as those that have worked in spaces such as Boston Scientific, FDA, and research hospitals. We're currently raising a seed round right now, as I mentioned earlier, of $2 million. We have over $1 million committed. And as I mentioned, an SBIR grant that we're looking to receive funding on with a score of 25. Our goal here is to change the paradigm of post-surgical adhesions by giving a technology that can prevent them in the cardiac use case, starting with congenital heart defect patients. However, we know from talking to surgeons that we can also use this product on other indications such as orthopedics, tendon surgeries, and OB-GYN such as hysterectomies and obstetrics or C-sections. So we're really excited to present our technology to you and would love to talk to you more about what we're doing at Karios. Thank you so much.
Greg, this seems like a game changer. Super exciting. As you mentioned, preventing surgical adhesions would be amazing for a lot of patients. Better outcomes at a lower cost would be my guess. I'm going to start with a couple of questions. You mentioned that your market entry point is with pediatric cardiac surgery. Woohoo! As a pediatrician, I love to hear that. And tell me why you're starting there. I'd like to learn more about that.
So in examining the landscape here and looking at other products that have been attempted in the abdominal and gynecologic space, we saw an opportunity with cardiac because number one, these surgeries are very expensive and life-saving. And for the congenital heart defect patients, specifically single ventricles, they have a staged operation set where they have a first operation followed by a second operation at 3 to 4 months. This will allow us to utilize a novel technique of non-invasive imaging to first assess the adhesions with imaging and then perform the reoperation to assess the product's performance versus no treatment groups in a small cohort.
You know what? That makes a lot of sense. A lot of sense. I know exactly what you're talking about. All right. So tell me a little bit more as well about the key differentiators of the Karios product compared to other products that are on the market.
Right. So the big players in this space are really two films, Sepra film and Interseed. And these are, like I said, things like saran wrap that you try to wrap around an organ or, pack into place in tissues. They're very difficult and they don't work very well and degrade rapidly. There's also viscous solutions that just don't do the job and can't translate to the cardiac space anyway because it drains. The secret sauce for us is our cross-linking chemistry is high efficiency, resulting in an integral network, which means we last 6 to 8 weeks in vivo, allowing the body time to heal. We also have that added tissue binder that in the liquid state coats the tissue when it's applied and keeps it in place so it doesn't dislodge like other sprayable products like Co-Seal and Duraseal, for example, when they're sprayed over large volumes. And the real key here is that we don't swell versus these other hydrogel products that swell 400 to 600%, which in a previous pediatric cardiac study caused tamponade.
Oh my gosh. Okay. So potentially bad outcomes with the other products as well.
Correct.
One
Serious
of the things
AEs.
one of the things that you mentioned actually leads into the first question from our audience. Are there cellular issues with the surface where the hydrogel is applied? What kind of data over time do you have on potentially this type of issue?
Yeah, so I've published implanting this in the sub-Q space, and we have non-published data in the IM space, but we have mice, rat, and pig data, oh my gosh, in the cardiac space. And what we've seen in all of these is that it's biocompatible, biodegradable, with no chronic inflammation response, and that we see a very typical response you would see from these benign materials, so to speak.
All right. And another question from our audience. How does your product affect wound healing, if at all?
Yeah, we did not see an impact. So that's a metric that we'll be measuring in that piglet study that I talked about, as well as a number of other key facets. Come by the table, and I'd love to tell you more about this great piglet study that we designed with our medical advisors who are chiefs of cardiac surgery across the country. But no impact.
All right. So another question around, would the product get immediately suctioned and or drained by chest tubes or drains, depending on where the surgery is, in a variety of different places?
Love this question. Great question. So our product gels in less than 3 seconds. So that's key. We tuned the gelation mechanism. As you saw in that video, you could see it gelling rapidly. You could see a little bit of the gel at the top of the rib cage or the entry there. And so we gel immediately upon contact with the tissue, curing and forming that barrier that protects it. And that's really key. And we don't dislodge or fluff off like other products.
And the reality is that product stays in place until the body's natural healing is that 6 to 8 week window, correct? Where the body's natural healing is taking place at that point.
So I'm not a surgeon, right? My partner is though, and then from talking with other surgeons, we understand the big window is that 4 to 6 week time period, which is why we invented a product that lasts 6 to 8 weeks, giving the tissue time to heal, the body time to reach homeostasis. However, it is important to note, this was a feature that Dr. Madani, our co-inventor, was so excited about, is he wanted to be able to peel it away if he had to. And you can mechanically remove it if you need to, as we show in our Nature paper.
All right. Very cool. So one last question, which I think is a great one that I had as well. So beyond cardiac surgery, what other conditions are you looking at? Where else could this be used?
So the two next areas that we've received a lot of interest are in orthopedics, specifically hand tendon surgeries. The surgeons in this space that I talked to, the hand surgeons, think that we can prevent that second surgery where you need to release the tendon. So they're really excited about that, and we're working on developing an animal model and a grant submission there. Another area that I'm very passionate about is we want to go after C-sections and hysterectomies. This is a really important area that affects hundreds of thousands of women in the US alone and can actually prevent them from having additional pregnancies, and the current products are just not effective enough. So in summary, we're going after CHD cardiac market. This will expand the ability to use it in other cardiac cases because of our non-invasive imaging, so we won't require the reoperation. Come by the table, I'll explain that in more detail. And then we're going to stagger SBIR grants and STTR grants for those other indications. So if you're interested in collaborating, please let me know, because it'd be great. Sorry, getting it all out there.
No, you know what? I love it. Greg, thank you so much for being here. I know Dr. Macaluso, who's asked a couple of our great questions tonight. He'll be at your table afterwards because that last one was one from him. So, Greg, thank you again for being here, and thank you for telling us all about Karios. It's really exciting, and again, potentially a game changer. Excuse me, in the field of surgery, right?
Appreciate the opportunity. Thank you so much.